So the third session will examine some new processing technologies and how they're being integrated into current practices. To kick us off, we have Andrew Stanley, General Manager of Innovation at Sanford. Andrew will tell us about the progress in their bioactives R&D. Over to you, Andrew.
Well, good afternoon, everyone. My name's Andrew Stanley. I am an introvert, and it's been 605 days since my last public appearance. So I say that because I'm a little bit nervous, mainly because if you know me, you'll know that I like to raise tricky topics. I like to start new conversations, and I think I'll do that today. But also want to follow that up by explaining how we're trying to remedy the situation that I think exists. So the situation really is that, you know, we as a primary industry, so this is not just seafood, this is a primary industry, there are challenges that we face as a primary industry commercialising science in New Zealand. We certainly need to have a thriving research and innovation sector. That is vital. If we don't innovate, effectively, we run the risk of becoming obsolete. It is incredibly important for growth, but also sustainability, having a vibrant economy, a resilient economy, and you know, value in increasing living standards really comes from innovation. We do have good science. We've seen a lot of good science this morning, and I know we'll see more this afternoon. And we have awesome people. However, the funding system that we work with in New Zealand does tend to lead to institutes chasing projects with predefined scopes. Now, often we do that in order for those institutes to survive. Industry is involved and it's asked to support, but more often than not, that engagement doesn't happen at the very start, and the science isn't necessarily completely targeted to a useful commercial outcome. Sometimes institutes control and value the IP highly, and the institutes themselves don't have the capability to commercialise. A lot of the time, really good science gets left on the shelf. So science and industry across New Zealand, across primary industries, is still fragmented, and we don't have mature pipelines from science to economic value, in my opinion. Broadly speaking, there are examples where we do have good pipelines, and plenty of examples where we don't. And if we are to develop better commercialisation pathways, they do need to be consumer and business led. We are a small country and we have limited resource. So the question really, the provocative question is that all of this that I'm saying here true for the seafood industry, for what we're doing here? And then what is our response to that? Machine, here we go. So I threw this together. Basically, it's a way of describing the fact that we have our innovation process and our science process here on the left-hand side that do come up with great ideas. They're somewhat collaborative, but there's a massive gap, a massive distance from that idea through to economic value. And some of the steps that I've listed there are a lot of the things that we as industry need to deal to before we can actually see commercial value out of opportunities. Of course, the funding does happen more often at the science stretchy end of the pipeline, but there is a massive and often long distance between concept and real value. I was just thinking over lunch about an analogous sort of situation. I sort of feel like if you're a soccer team and you had the goal at the other end of the pitch, we've got a lot of fancy moves down the defensive end, and we go to pass the ball up the field to the ones that are going to put it in the goal, and we're a little bit absent, or we don't have a plan, or we're disorganized, or you know what, the goal is a hell of a lot further away than we actually realized. So in 2012, roughly, this is what we did in Sanford. They said, get back in your box and start thinking outside it. And we've moved it a long way since then. Our current team is, we're based primarily in Blenheim, but we do have team members around the country. And we're seeking to really boost our own capability, but also the industry's capability to manage that pipeline of creative ideas, to provide some coordination, collaboration, innovation, be involved across a range of platforms and projects, to leverage our vertical integration and scale, and get support and seek support from investors to drive home some of these initiatives. So take them off the shelf and deliver real value. So I've put in the bottom there, meaningfully contributes to maturing the seafood industry innovation pipeline. And I think this has been missing. So for us, what that means is we're in the process of constructing a marine extracts innovation centre at the top of the south. This is in Blenheim. And this is the state of play. It is due to be completed, the base build completed by the end of July. And we'll have some operations in there in September. Building to a staff of 48. From our current little extract site in Riverlands of 340 square metres, it's a considerable expansion. It is an expansion in capacity of existing processes, but it's also a site where we can centre our innovation activities, where we can conduct scaled up extraction processes of a range of marine products, only marine. We're not doing anything outside of that. We are introducing new technologies. And with this in mind, with the centre being there, we have this opportunity to broaden our involvement and our degree of collaboration across the innovation ecosystem. I think more than anything else, this is about strengthening that pipeline. You know, in an environment where we have really limited processing capacity in the industry for novel products, this is could potentially fill quite a vital link in that chain. And the other work that we do from the centre will support that value chain as well. So the site itself, just to give you a sense of scale, the NZAC factory in Blenheim, 340 square metres, is smaller than our administration block here, just to give you an idea. I won't bore you with the full video, but you'll get a sense that it's a modern facility, it's well thought out. There's an integration of sort of lockdown processes and wheel in, wheel out, flexible processes. There's space for innovation, there's space for lab testing. There's space on the site to grow. It's a facility that represents really a vision from many years ago, and it's there in the flesh now, and it's extremely exciting. It's not until you walk into the site, so this is cool to look at the video, but when you actually walk in the site, like I did this morning, and you have a look around, it's incredible the scale that we've got. That's really neat. We're not going to be confined to 50 or 60 square metre or 100 square metre processing rooms at other people's places. We're going to be doing this ourselves. We're not going to be able to do absolutely everything you can conceive of, but there's a willingness to do what we can to take opportunities that come out of our science infrastructure, our science ecosystem, and push them through into commercial products, take them to market. So that should be under 10 minutes, and that is me. Thank you.
Right, so our next speaker is Dr. Owen Catchpole, team leader and chief engineer of integrated bioactive technologies at Callaghan Innovation. Owen is the leader of Cybermarine Research AIM 1.5, which will test the flexible processing concept in practice. Owen, just to be clear, you have 15 minutes for your talk. So the other two talks have 10. In this session, you have 15. You can start.
Let's just hope I've chosen the right screen.
Looks good.
Okay. Actually, let's go back a step. So, yep, title of my presentation is Extraction of Lipids from Partially to Fully Dried Marine Biomass Using Supercritical Fluids. And let me say right up front, we are looking forward to install some of our technologies being installed in the new factory you've just heard about. So a big disclaimer while I'm doing the presenting of this talk. In fact, Joel and Morel has been doing most of the work and working with the rest of the team here to come up with the results. So a few quick words on what Callaghan is doing in Cybermarine. So probably the two biggest tasks are developing of these new processing technologies and working with Planted Food and the others on the sequencing of the technologies. Particularly, our part is particularly focused on the lipid extraction side of things. Then our lipids and natural products team is doing a huge amount of work on both the detailed lipid composition of the exemplar marine biomasses and also the samples that we produce from processing. And to some extent, a bit of development of analysis technologies that may become online or near online. And then Cynthia is doing some work on isolation of some quite unusual enzymes from fish. Later on in the program, we'll also be working with Planted Food on how to fractionate some of the crude extracts we get and to make them into sort of proof of concepts type final products. And then the last and somewhat scary challenge for me is how we put it all together. So actually, yeah, I meant to say, let's go back a step if we can. Oops, now I've gone too.
Yes, so I meant to say our extraction technologies are largely based on dimethyl ether and supercritical CO2. The next slide gives a brief a comparison between the two solvents. Carbon dioxide is already well known, being used for producing green shell mussel oil. Dimethyl ether, to some extent, is slightly newer, although I've been working with it for an awfully long time. Probably the bits of the table to look at the most is in terms of its flammability. It is flammable, unfortunately. We have done a lot of work to get it approved for food use in New Zealand, Australia, US, and others have done in the EU. But the big advantages are that it can extract these polar lipids or phospholipids, and it can also extract wet biomass. So we can do production up to a 200 litre scale in New Zealand, whereas the CO2 can be done at a very large scale, 4 by 1,000 litres at final extract state. Okay, so just going to show you a processing sequence for the extraction of biomass as originally envisaged. So the material, the fish is minced, we dry it, we extract it with supercritical CO2, and then you get a nice sort of little pile of hopefully high omega-3 lipids. What we've also been investigating recently is whether you can replace the mincing step with mechanical type separation, and where you can use something like a screw press or a beehive, which gives you a kind of a flesh fraction and a bones and skin fraction. And while they look almost identical when they're dried, they're actually pretty different in terms of composition. So yeah, just a little bit more on that. the mechanical separator can separate it into flesh and meaty skin, meaty to skin bits. And the preliminary work we've done so far already shows that the bony and skin bit has got a lot more collagen, peptides, and mineral than the flesh. So what have we achieved from our baseline extractions on dry biomass? So they've been done on whole Jack Mackerel, WJM, or lean fish, Hoki, frames and heads, and of course the green shell mussel. What you can see if you look hard enough at the graph is the yields from DME and CO2 are more or less pretty much the same. That's the yield on the y-axis. But the one where there's the biggest difference is green shell mussel. The yield from DME is virtually double that of CO2, and that's all due to co-extraction of phospholipids. So this engineering presentation wouldn't be complete without a table that's hard to read. If you just focus on the blue and green, which is EPA and DHA, not terribly exciting from the Jack Mackerel or the Hoki heads. I mean, you know, goodish kind of levels, but nothing to get too excited about. When we go to this slide here, this sort of really got us thinking. One of the things I've noticed with DME extraction and using organic solids is that we never quite get the full extraction of phospholipids. And it seems to be caused by, I guess you could call it over drying of the biomass. So the real question for me then is, can we optimize the extent of drying followed by DME extraction to achieve almost complete yield of the phospholipids? So again, to reiterate, this is what the normal processing sequence looks like. The mussels are separated from the shells, they're minced or homogenized in some way. They go into the freeze dryer, they're dried to a powder, dried and ground to a powder, extracted with supercritical CO2, and then you get a reddish orange looking oil, which is blended with a different type of oil to give you a classic lipinol-like product. What we're doing instead is looking at whether we can partially freeze dry the biomass, extract it with DME, and then get our extracts. Oops, let's go back a bit. So the steps that we want to optimize are how you mince the mussels so that you don't get degradation of the phospholipids due to the phospholipases there, the extent of drying, and the DME extraction of the partially dried biomass. I promise I won't go too much into the detail of this particular graph, but the red line shows a typical temperature profile for freeze drying. Starts off with the material being frozen on the tray. You push up the tray temperature to sublimate as much of the water as possible, and then you reach a point where the primary drying is finished. You push up the temperature and you get the rest of the water off. Now that last stage of the freeze drying is both highly energy intensive and pretty low efficiency. So if we could stop the drying at the second plotted line, you could have a much shorter drying time and a much greater throughput. So does it work? And this bottom graph shows that yes, it does. So the key to look at is the green one. And if we go to the right of the graph, it's 75% moisture content. That's the typical water content of green shell mussel. You can see our residual lipid content in the biomass after DME extraction is about 1%. If you go right to the left-hand side, the residual lipid content is more like about 3 or 4%. So somewhere in the middle is about the optimum of what we want to achieve. And interestingly, our work sort of replicates what Daniel had found, that the lipid content was sort of much higher than what the literature suggests for green sugar mussels. Let's go to our next slide. These curves show on the left-hand side the total extract from the green sugar mussels. So at the top graph here, you're extracting an awful lot of water and not much lipid. The bottom graph, which is dry biomass, you're extracting all lipid and no water. What's interesting also for us on the right-hand side is you can see the water. This is an extraction rate curve, that the water and oil, at least at the start of the extraction, are being pulled off at more or less exactly the same time, and then they both start to plummet off quite rapidly as the extraction goes on. But most importantly, what do we actually get? So again, we've given the omega-3 content data, and you can see that at all the moisture content levels, EPA and DHA are pretty much the same. So that's the first test passed. It gives you the extracts you get have got the full complement of omega-3 fatty acids. And then in our next slide, possibly the most exciting one for me, is that extracting the wet biomass gives us not only the total phospholipid extract, but a virtually unchanged phospholipid profile from what's in the feed material. So the phospholipid profile of green sugar mussels is extremely complex. So I'll only show you 4 of the compounds that are there. The phosphatidylcholine, that's the most kind of standard sort of phospholipid in marine biomass. Then you have the second one that Mikhail touched on, E-plas or ethanolamine plasmalogens. They're a pretty high concentration in green shell mussels, and as he noted, they're a possible anti-Alzheimer's therapy. The ceramide aminoethyl phosphonates, they're a special class of phospholipid that's got a direct carbon to phosphorus bond. And some recent work has shown that they've got potential for being a dietary cosmetic, sort of where you take it in your diet and it improves your skin barrier function. And kind of the sort of lipids that you don't want so much, look at the lysophospholipids. We've shown that they've unchanged due to our processing. So what have we found in conclusion? That's from dry biomass, you can get pretty good extraction of fatty acids. Mechanical separation is potentially a good alternative to mincing and homogenization. If we're trying to get different fractions to work on or make our job a bit easier. But the DME yields of phospholipids were incomplete from the fully dry biomass. We go to our partial drying and DME extraction technology. We can get full extraction of the phospholipids, and we can improve the efficiency and energy usage in the freeze drying as well. And the phospholipids themselves are fully extracted with no degradation. So that's very promising for us in future work. We'll look at taking that phospholipid fraction and try and fractionate it further to get these individual phospholipids. And that, despite my unexpected shutdown on my computer, is it. Thank you for your attention.
Okay, next up we have Bodhi Betcherman. He's a research technologist here in PFR Nelson. Take it away, Bodhi.
All right, hi everybody. I'll just get us in full screen mode. Yeah, my name is Bodhi. I work in Sue's group with largely in the pilot plant in the marine products group. Today I'm going to be talking about how we're going to be integrating different processes in a cascade to deliver multiple products. So Current processing methods tend to focus on making a single product from a natural resource, such as fillets from fish or oil from green shell mussels, with the remaining material largely being an afterthought and not a lot of consideration being given to the preservation of the valuable components that are present in them. The focus of the Cybermarine program is about making more high-value products and therefore more profit from the same raw resource. To achieve this goal, we need new approaches to the processing of the raw material. So marine organisms have many potential products that can be extracted from them, and with careful process design and considered staging of the unit operations, it will be possible to expand the range of products that can be made from a single species. So some examples of these additional products that we can make from these marine species include things like digestive enzymes. Now, these have a lot of value if they're isolated and purified just in themselves. They have value in industrial processing applications and also in medical applications as things like digestive aids. But the real value to be gained if you can remove these at the right time in the processing is that by taking these digestive enzymes out, you'll be reducing or stopping the breakdown of other valuable components within the extraction pathways. Other areas of interest are lipids. So obviously there's lots of other sort of fish and shellfish oils on the market. And as Owen's been talking about, there's a lot of interest, well, there's the new focus is on trying to tackle those more challenging polar lipid fractions that are present in the species. So I'll talk a little bit more about the work we're doing in this area shortly. Other products are obviously the collagens or extracellular proteins like collagen, and things like you can extract the muscle proteins like actin and myosin to make food products. And then there's also you can use digestive enzymes like proteases to pull out some of those inaccessible proteins from the material to create peptide mixtures. So we have a number of technologies at our disposal to try and achieve this cascade and this pathways. We're interested in using techniques that are largely aqueous and quite gentle to avoid damage to things that we're interested in. And so these are things like using centrifugation, filtration, potentially adding enzyme inhibitors early in the process to keep those enzymes under control. We can use selective precipitation techniques by controlling the pH or the salinity or the temperature and adding flocculants. And we also have a lot of membrane, different types of selective membranes that we can use, like ultrafiltration and nanofiltration membranes, which have a chemically modified surface to achieve good separation of solutions. And then to tackle those tricky polar lipid compounds, Owen's already talked about the work that Callaghan's doing with supercritical fluid extraction and dimethyl ether. But plant and food research is looking at aqueous ways of tackling this. And so some of the work we're doing is using NADEs, which is natural deep eutectic solvents. And these are combinations of naturally occurring compounds, if they're used in the right combination, form these ionic solutions that can help to pull out these polar lipids. And they can be used as an alternative to organic solvents in some cases. Another approach we're using is using surfactants. So these are compounds that have a hydrophilic head and a hydrophobic tail, and they're essentially sophisticated detergents. We have a whole suite of different types of surfactants that we can use, and hopefully we can use these to help pull these molecules out of solution as well. So the way we're going to go about this is starting in the lab, we need to develop a whole suite of different analytical techniques. And this is important so that we know what we have and where it's gone, and make sure that it's still that the molecules of interest still have the function that we're interested in. And this is not a trivial thing to set up all of these assays and analytical techniques. And so there's going to be a lot of work at the start getting this up and running, but this will set us up for the next stages. Once we've developed these processes, we can design process flows that will fit these extraction techniques together to try and target the molecules of interest without damaging downstream molecules in the processing. So then the next stage is to scale that up, and that's sort of my area of specialization. And we'll be taking these things to the pilot plant and running these operations at pilot scale. And then once we've got ironed out all the kinks and worked out where all the issues are, it's really important to run things at pilot scale, because transferring a process from the lab to industry, scaling it up always throws up a whole lot of issues. So it's quite good to test it at pilot scale, and after we've established good processes, we can take them to our industry partners in factories or rendering plants and run these processes at a much larger scale. So ultimately, our goal is to create an integrated biorefinery, which we can run at a pilot scale, and we pull together all of the work that's done across Cybermarine to make these multi-product cascades. So yeah, hopefully you guys can all see the opportunities that are available, and yeah, I think we're doing good work.
Thanks, Owen. I see Sue has a question.
Yeah, that's right. So my this is not really muted, and since I foisted this on Bodhi, I thought that I better come and just do a little bit of an extra explanation. I don't think it came across particularly clearly about the difference between what Plant and Food is doing and what Callaghan is doing, and how the two approaches complement each other. So Owen is looking at things that are dried and partially dried and extracted using supercritical, whereas we're taking an entirely aqueous approach. So the thing being that we don't know whether what we're doing is going to work and whether it's going to work at all. And so we're hoping that by the time we get to the end, we'll be able to choose the best processes from the whole thing and put them together to optimize the process. So we're not using any solvents at all, and we're not drying at all. So Owen is looking at energy improvements in a different way to us. We're looking at energy improvements through not drying until we get to the very end, and taking things out, always using aqueous. So it remains to be seen what's going to happen, and it'll be in RA 1.5, which Owen has to run, where we get to put it all together and work out what of all the things that we're working on are going to pull out the things that we want in the best possible way. And so a lot of the focus that we have is on the order of extraction. And so we're all developing lots of different unit operations, but the key to them is working out which one to do first, so that you take out things which destroy, and Bodhi talked about this, so we're very much taking the enzymes out first, and then that leaves us with a much more stable raw material for subsequent reactions. So that's all. Thank you.
Yeah, that was a pretty good addition there, and it's quite likely that we might be doing the lipid extraction step, as Sue says, after other stuff has been pulled out, and so it's helpful to know that we might be able to extract that from wet or partially dried biomass.
Okay, Well, thanks for that clarification, Sue and Owen.